Hello everyone, today the topic is about enzyme substrate binding, and we can understand this enzyme substrate binding with the help of three models: lock and key model, induced fit model, and a substrate strain theory. We will understand all these three models one by one. First is lock and key model. Now this was proposed by Emil Fischer, and in this model it was told that the shape of the substrate and the enzyme active site both are rigid, fixed, complementary to each other to fit together like a key in a lock. The substrate is compared with the key and the enzyme is compared with a lock. Both of them are very much rigid. Both of them are rigid and both of them are fixed in their structures, and they fit or they complement to each other in their structures so that they can bind like a lock and key. Next is the induced fit model. Now this was proposed by Daniel Koshland, and it requires active site to be floppy and substrate to be rigid. Unlike the lock and key model where both substrate and enzyme were fixed and rigid in lock and key model, but induced fit model, you can see here the enzyme site or the active site of the enzyme is floppy. Now because of this, whenever the binding of the substrate comes and binds to the active site of the enzyme active site will undergo a conformational change, and it will change according to the shape of the substrate. Now this floppy site of the enzyme will change its shape according to the substrate. So that is why this is called as induced fit model. Now the enzyme changes the shape similar to that of substrate in the transition state, and during transition state additional bonds may be formed or broken, leading to transformation of substrate into the product. Next is substrate strain theory. Now in this as the substrate flexes to fit the active site, the bonds in the substrate are flexed and stressed, and this causes changes or conversion in the product. Now here the active site of the enzyme is floppy, and to fit to this active site of the enzyme, the substrate will undergo some strain, or it is flexed and stressed, and this substrate undergoes flexion or stress, and this causes changes or conversion to the product. So this is how the substrate is, and it undergoes some change or stress in to get fit into this active site of the enzyme, and because of this it undergoes conversion into product. So to compare, this is the lock and key model in which both enzyme as well as substrate are rigid and fixed. Both enzyme and substrate are rigid and fixed. And next is the induced fit model where the substrate is fixed, whereas the enzyme or the active site is floppy, and the substrate will fit into the active site. Once it comes and fits to the active site undergoes conformational changes so that it binds according to the size of the or the shape of the substrate. Next is the substrate strain theory where both of them are floppy or flexible, and the substrate will undergo a strain to fit into the active site of the enzyme, and because of that strain the substrate will get converted into a product. So these are the three models which explain about the enzyme substrate binding or the enzyme substrate complex formation: lock and key model, induced fit theory, and substrate strain theory. Thank you.